I am Chris Chou, and I teach at Longmont High School in Longmont, Colorado, and I'm presenting this session on behalf of 3D Molecular Designs. So I have been using products from 3D Molecular Designs for the last about 7 years, and it really started from me attending a teacher workshop over at Milwaukee and really learning how to utilize the power of these modeling kits. So, all right. I do teach in Colorado. So Caitlin, when you're mentioning the fire tornado curriculum, I am unfortunately very acquainted with fires here. Last night I was telling Chris, who is my co-moderator here, that there was a fire that started like 30 miles from where I live near Boulder, Colorado. And it was high winds and a power line got downed and started a fire that just spread like crazy. And a lot of communities just west of me were starting to get evacuated. There's 900 households that were, and I was like watching late last night, like, am I gonna have to be evacuated? And if so, can I show up still this morning for the session? So thankfully we got some rain and fog this morning, which has, and our brave firefighters. So that's beat back the fires. And so able to be here with you all. So thank you for letting me be part of your morning. Okay, so our session today is using models to make connections between DNA and chromosomes. And we're going to be using some kits that hopefully got sent to those of you who are who pre-registered, but if not, don't worry. You'll just kind of follow along with us and kind of do some thinking and responding as well, because maybe you might be teaching in a remote situation where you have the materials and your students don't. And this would be a great way to be able to sort of elicit conversations with your students and to demonstrate these processes that we're going to talk about today with these models. So go ahead, next slide. All right, so I'd love to hear more from those of you who just slipped in here and came to join us. Where do you live and what grades or subjects do you teach? And I know there's a mixture of middle school and high school teachers, some maybe in science education. So feel free to type that in the chat. Where do you live and what grades and subjects do you teach? Just for me to get an idea of who my audience is here this morning and maybe speak to, you know, middle school adaptations for this activity versus all the way up to maybe the AP or college level. Okay, from LA, San Dimas, Sacramento, pre-service teacher. Welcome, pre-service teachers. I think there's a couple of you guys here. I think this is a wonderful way to like connect with fellow educators, just kind of see what's available for you. All right, Stockton, Elk Grove, Santa Clara, San Jose. So like, looks like up and down. I know there are some from LA, so welcome. All right, so our workshop goals today are to model chromosome structure and anatomy. We're going to take on the very large task of comparing and contrasting mitosis and meiosis, mechanisms that contribute to genetic variation, and at the end to take a look at how we can use these models to show at the molecular level how genetic conditions can be inherited and connecting that with Punnett squares. So you can use it through a variety of units and topics that you'll be teaching either at the middle school all the way to the high school level. So next slide. So as I mentioned earlier, I have been utilizing products from 3D Molecular Designs for the last 7 or 8 years. And through the course of these 7 or 8 years, slowly accumulated some of the different kits that they have. The ones that I'm showing in this picture, are my actual students from last year before we went completely virtual. And you can see that they're working in groups. There's smiles on their faces. They're also laughing at how Mrs. Chou is just a paparazzi and is like taking all these pictures, but that how they love getting their hands on these physical models. And there's a power in communicating with your group members, eliciting your ideas. We also spend time with these models, taking a look at what's useful about these models, but what are some limitations? And so they also do a little bit of design thinking of how could we modify this model to make it represent better, in sort of the scale of some of these processes, so that it shows better, what's truly involved in these really complex processes. Okay, so next slide. The one kit that we're going to talk about today, the main one, the classroom version of this kit is called Chromosome Connections. And you can see on the picture on the left is I have 6 full classroom kits, and I usually have these tables in groups, so where they have 4 to 6 kids, and each group gets their own whole kit. And I kind of organize my stuff, as I call it, stuff management, with trays, and then each group gets a tray with some pre-assembled chromosomes. You can see those yellow things are kinetochores and spindle fibers, and those are part of the classroom kit, but we don't have them in our student modeling kit, but I'll show some, I'll give some suggestions of how you can modify that for home. But you can see here, my students from last year, sitting at their tables, they are modeling the process of crossing over with meiosis, and we're going to get into that and do some of that today. Next slide. So for those of you who pre-registered, and you can also request this student kit. This is the Chromosome Student Modeling Pack. And you can see it has fewer chromosomes than the actual classroom set. And there might be some pre-assembly involved here. So if you have this kit with you, and I wonder if some of you could just maybe type in the chat like, yes, I have this kit, or not yet, don't have that yet. That way I can know that. And then Rachel typed in the link to be able to get your own kit too. So that'll be fun for you to have. So I would highly recommend that you go ahead and register and get one of the free packs. So this kit comes with some things you might need to pre-assemble. These things aren't critical for today, but just so you know, what you may need to do is it comes with, and if you can see my video, not sure if you can see my video, but there's a like 4 cutouts for this connectors that we'll talk about, and then a couple of handouts with creating a giant Punnett square. So that might require some cutting and taping right there, but not critical for today. Really, those foam pieces that you have, if you have them in front of you, punch them out, and we're going to assemble some things. So next slide will show what I would love for those of you who have the kits in front of you, to pre-assemble them. And if you don't, that's okay. I'm going to kind of describe how you would have students put them together. So you're going to need some of these parts. If they're cut out, great. If not, you can just kind of lay your foam pieces on there. You can see on the left, there's actual large chromosome structures and connected to some linker parts. And I just want to mention that these linker parts don't come attached to the paper. So I just used Elmer's glue and glued it in there. And then you can attach like other things to it. Same thing for the other side, is like you'll glue on those linker pieces. So what you're going to put together in the middle there are some of those nucleotides that you're going to punch out. It really doesn't matter what sequence you have. So just kind of put a few in there to demonstrate. From this graphic, we're going to see all the different ways you can represent DNA. So this is just different ways you can represent DNA. I think the powerful thing about this kit is this linker portion in the middle. That's the paper part, actually. And it really connects for kids. We'll see in just the next slide what's happening there. So I'll give a couple of minutes for this. For folks, like once you've got this assembled, go ahead and type in like I'm ready or something like that in the chat. That way we can be ready to move on to the next part here. All right. Hopefully you are all able to see Heather Ryan's camera image. If you go to, I guess, the grid view, you might be able to see her grid video that she had was building that. Trinity, I think were you working on that over there as well? Great. Did you have like a glue stick in your hand? You're gluing things together?
No glue stick because I knew it wouldn't dry and then I'd be like sticky glue all over my fingers and like, going crazy.
Okay, great. All right, so Heather's there. She's working on that, so you can see her like putting things together as we're going along. I hope no one fell asleep. I was told in feedback that the timer that I had earlier was too jarring and it had a really annoying blaring sound at the end, and so I picked a soothing one, but hopefully no one fell asleep this morning. All right, so Michael, do you want to talk about some of your thoughts? You said this is cool. Some of your thoughts as you were building this so far?
Yes, the fact that you have hands-on models that the kids can really not only visualize anymore, but can really work with, is just a potential game changer. You know, because most of things are not is so abstract when you're talking about DNA and RNA and all of that. But to be able to have these models that you can really assemble is just amazing. Yeah, so thank you for that.
Absolutely. Thank you for that feedback, Michael. I'm really glad that you felt you had that kind of realization too. I think when you introduce these to your students, they're going to come away with, wow, this is something that I can touch and see physically, even though it's at a molecular level. So we're going to take a look at this slide here of the organization of eukaryotic chromosomes. And so generally, the models that we're going to be looking at here are linear chromosomes. Those are eukaryotic. Prokaryotes have the circular double-stranded chromosomes. One model that 3D Molecular Designs has, that there was a session yesterday about this DNA model. And I don't know if you can see it here. So it's kind of fun. Actually, this is the first time I played with it. I haven't had this in my classroom. And I got a hold of one of these kits and I was like, oh, why have I not used this before? And part of it is I didn't know what to do with it. I assembled parts and things kept falling off. I had no idea. Anyways, this is the benefit of these sessions because you can see how these models can be utilized in the classroom, and a teacher can, talk about pitfalls and how to manage, materials and students too. So this is, the double-stranded model. I love this one in particular because you can see with some arrows the directionality of each strand of the DNA, as well as the base pairing of A's, T's, G's, and C's. And then you can see how it can rotate and coil up. So that's at the basic level, is the sequence of nucleotides. And then when you think about DNA being really long molecules, it needs to be organized in a cell. And so they're organized by the condensing of the DNA molecules, and they're condensed around these proteins called histones. And that DNA wrapped around the histones in these groups of 8 histones form a unit that you're zooming out called a nucleosome. 3D Molecular Designs also has a nucleosome modeling, which is great because you can show the packing and unpacking of DNA for transcription. So thinking about how can you turn off transcription with methylation of DNA or histone acetylation and the unpacking of the DNA molecules. And then as the chromatin is further, the DNA is further condensed, it condenses, into chromatin, which is most of the DNA within a cell is usually at that stage. And then finally, at the very bottom is showing you chromosomes are visible under a microscope when they are getting ready, when cells are getting ready to divide. So those chromosomes are totally packed up together and the DNA has been replicated at this point. So what you're seeing at the bottom here is a duplicated chromosome. And so on the next slide, as you discuss, this level of organization of DNA from the very zoomed in level of the base pairing and nucleotide sequence all the way out to the visible structure of the chromosomes, you can also compare that with what you just constructed. So what are some things that you guys notice on this particular model that you have created or assembled together that shows that diagram that you guys are seeing on the screen there? So go ahead and type that into the chat. What are some visible things that you're seeing in this model versus the diagrams on the screen? Okay, so hopefully you're able to make some comparisons there. Let's see, Chris, would you mind kind of using your mouse to hover over some things? Okay, Trinity, thank you for typing that in. You see nucleosomes on your model as well. Okay, so Heather, I'm sorry, Chris, if you could, yeah, right there, those are the nucleosomes. And then if you could point to the nucleotides, you can see that on your model right there. And then the further condensing of the chromatin. And then finally, the duplicated chromosomes, or not yet, that chromosome structure. Okay, great, thank you. All right, and Diane said she always liked the scaling feature. Yeah, so I think with this, both the student modeling kit, you're seeing the example on the right side comes with colorful nucleotides, the A's, T's, G's, and C's there. And then this was the connector kind of graphic. In the classroom version, the chromosome connections kit on the left, the nucleotides don't come as the different colors. It does come with all red, showing maternal chromosome, or all blue nucleotides showing paternal chromosome. There's a reason for that, and we'll show you later kind of a fun way to be able to show how crossing over occurs. And it's more helpful to have the blues and the reds with that. But the student kits come with these colorful rainbow colored nucleotides. Okay, we're going to zoom out now, and we're going to start to talk about cell division with mitosis and meiosis. And so we're going to go to the chromosome level now, and we're going to imagine there is a cell now that we're working with that has 4 chromosomes in this cell. So if you have your kit there, go ahead and punch out all the different parts and assemble the chromosomes that you're seeing there. So you're going to need to, for part of this, to take off some of the chromosome foam parts off of your scaling graphic and actually assemble, use them to assemble into those 4 chromosomes. They don't necessarily have to be made exactly looking like those on your screen, but it's helpful because it gives you some examples of what these chromosomes look like. So I'm going to give you a few minutes here to do that at home. All right, so now that you've assembled your chromosomes in this fictitious cell here with chromosome number is 4, let's take a look at what some of these parts are. So this is where you can have your students have these models in hand, and you can have them like point to the parts as you're talking about other different components. So on the next slide, you will see that the ends, let's start with the ends. These ends are called telomeres or telomeres. And they are repetitive sequences that are non-coding at the ends of the DNA to really protect DNA as it goes through multiple cell divisions. And every time they're replicated, we know that DNA gets shorter and shorter. So the telomeres protect the ends from this DNA erosion here. Getting to the coding sequences. The different colors represent paternal and maternal chromosomes. So the ones that are blue are the chromosomes you inherit from your father, or the male contributor to the DNA there of a diploid organism. And then the red ones represent maternal chromosomes. And so you'll notice that there's 2 copies of each type of chromosome. You can tell by their length, also by where their centromere region is. So that next, click on that. Yeah, so the chromosomes that have 2 copies of each type of chromosome, those are called homologous chromosomes. So they contain a variety of genes on each chromosome. And so you'll have that same corresponding gene on the homolog there. So every chromosome, as they condense together, will have these regions that look constricted called centromeres. This is the place where during cell division that kinetochores, proteins will attach to this region, and then spindle fibers will attach as well. So in the big classroom kit, you will see that it's kind of fun here. You can attach these kinetochores and the spindle fibers to them to like move these chromosomes. But for the students, this is the region where you can have them just use string at home and kind of tie it off in the middle here at that centromere region to move the chromosomes as well. So that's kind of an adaptation for home. So you can see that these chromosomes with the centromeres don't necessarily have them in the same spot. So this is a chromosome where the short arm is called the P and the long arm is called the Q. This is more like above, the smaller part is on top and the longer part on bottom. You can also have the centromere, which divides the top and bottom into equal lengths. So these chromosomes have a variety of locations where those centromere regions are. Okay. So as we take a look at all the different chromosomes in a human cell, because your kids are going to care about their own cells more, right? You can see that, and this kit also doesn't come with 23 or 46 chromosomes. That would be a lot. But it's a good representation of the different types of chromosomes we have. So you can see here that we have 23 different types of chromosomes. Chromosomes 1 through 22 are considered the autosomes, or and then chromosome like set 23 is the sex chromosomes, and with the X or the Y at the end right there. And so this karyogram is arranged in a way to depict the chromosomes. That banding is just the staining that happened with these chromosomes, and they're organized in a way that makes them easy to visualize. And so that they're from the longest all the way down to the shortest, little cutest chromosomes like 22. And then the Y is kind of a little shorty chromosome too. And then there's the X, which is a lot bigger. So that's a representation. But a lot of times when we take pictures of chromosomes, you're really going to see them in as homologs, like these pairs, because you really have a total of 46. So you're going to have 2 of each type of chromosome. And then the sex chromosomes, you can either have X and Y or 2 X's. And so that makes it a total of 46. And if you take a look at the picture of actual chromosomes in a cell, this is a karyotype now, and you can see them all lined up. And what would you say is the sex of this individual from this karyotype? Type that in the chat. And how do you know? How do you know? All right, excellent. Okay, so everyone's typing in. It's a female and XX and no Y chromosome. These karyotypes are great to be able to show your students, to have them see if they can tell based on the karyotype, is it female or male? And sometimes, if you see 2 X's and a Y, what is happening there? What characteristics might they have having an extra X chromosome? So a lot of different questions that you can ask your students, have them think about these things. And we try to be careful as we talk about gender identity, we're trying to go away from saying like girl and boy and trying to use more female and male. And so I know that's something language-wise that I've kind of changed a bit in my classroom to really just use female or male in that case. Okay, next slide will show you in the context of where are we seeing, how are we taking the pictures of these chromosomes? So this is a diagram of the cell cycle, the life cycle of a cell. And we usually, you want to start off in the graphic at G1, And you can see as it goes around, there's G1, S, G2, and then the M phase. And those are the 4 main phases of the cell cycle. And, oh, I see that, is it Elisheva had written, I use biological male or biological female. I appreciate that, right? Because of gender identity, and I do a great activity. This is a side note on gender, gender identity, or at least gender determination of athletes. There's a great like HHMI activity with that. And it, in terms of determining gender, very complex, so many different factors involved in that. So, right. Okay, so with the cell cycle, G1 is the phase where cells are doing their normal everyday activity, where there's transcription, translation happening, the DNA is being utilized for cell processes. And then after G1, if the cell's given the signals to divide, it's going to go ahead and go through DNA replication. So that is the S phase, DNA synthesis. And then after passing through the S phase, we're going to have a series of checkpoints of making sure the DNA was copied correctly. Are there any mistakes that have to be fixed? And then the cell's getting ready to divide through G2. And then the M phase is mitosis. Of course, we're going to take a look at that today, the different stages of mitosis. But going back there to that graphic, the DNA gets replicated in S phase. And then the pictures of the chromosomes are taken during metaphase in mitosis, because that's when the chromosomes have condensed in prophase and they're lined up at the metaphase plate in metaphase. And so that's the ideal time to take the picture. So it's important to let your students know that that's where those pictures of those karyotypes are taken, and that when those chromosomes, those pictures are taken, they're seeing something like this, and they look like flies. So this is a chromosome replicated, is what I like to say. So I like to say, this is a chromosome, this is a chromosome replicated. So that the pictures that you're seeing with 2 chromosomes in the karyotype is not this, it is actually this, chromosomes replicated. And you can actually see in the images, like there's a slight gap between the 2 sister chromatids. So that's a key misconception point that students have. And this is great where these models you can use to say, I have my students actually hold this up and go, okay, everybody, this is a chromosome. This is a chromosome replicated, so that you're using that same term for both of these things. Okay, going to the next slide, we're going to go ahead and do the S phase, or DNA replication. So take your original chromosome and replicate it, make an identical copy and have it next to your original. And give you a couple of minutes to do this as well. Okay, so you started off with, this is a chromosome. Now this is a chromosome replicated. So let's take a look at the different terms that we use to describe these different parts. And on the next slide, you'll see that this is a chromosome, but this is also a chromosome. And they are still, originally, these were homologous chromosomes. And after they've been replicated, they are still homologous chromosomes right here, okay? And then, but how do we distinguish these 2 little parts that, are identical copies of each other? Well, we call them sisters. So those are identical sister chromatids. Why they're not called brothers, I'm not sure, but we name boats after women and cars, so why not sisters? So, yeah. So these are sister chromatids, part of a replicated chromosome, sister chromatids, part of this replicated chromosomes, and together they are homologous. Okay, so we're going to take a look at this next graphic right here, this is showing the different stages of mitosis. And mitosis is needed by cells for reproduction, for growth, for repair. Like you think about skin as it sloughs off every day, you need to be replacing those layers of skin. And so you need to do mitosis. And so this is at the point where I talk to students about the terms mitosis and meiosis, and I kind of mispronounce meiosis and call it meiosis. And maybe some of you might as well, just kind of a little mnemonic device for kids. I say mitosis makes more of my toes or my toe cells, and meiosis is a process that makes a mini me. So you're going to need sperm and eggs for that to make your mini me. So that's that process. So, so I'm finding it really hard this morning to call it meiosis because I usually tell my students this is meiosis, making a mini me. So we're doing mitosis. This is going to make more of my toe cells. I don't know if how many of you use that too, but kids like silly things like that. And okay, so these are the different stages of mitosis. So prophase is where this graphic is kind of nice because it shows 4 chromosomes in the cell, and you all in your student kits have 4 chromosomes as well. So you can see how they have condensed in prophase. Metaphase are all lined up in single file order along the metaphase plate. And then anaphase, those sister chromatids separate. They say goodbye to their sisters. They're identical twins. And then telophase or telophase is that final stage. A couple of points is that the classroom kit allows you to be able to put these kinetochores and spindle fibers. So the kinetochore is like that protein grouping right there that connects the spindle fibers to the centromere. And you can use this to separate them and do anaphase like this. Also in prophase is when those spindle fibers start to like organize, and then you can attach them on. So there's a little bit more that you can do that's not exactly shown in that graphic that you can do with these foam parts. For students at home, this being a student kit, you can just say, grab whatever string you have, or even cut fabric or something, and tie it off and make this your spindle fibers to be able to physically move your chromosomes. And one thing I would recommend is you could have students at this point take pictures, since they all have their devices, of each of the stages and label them, put them into maybe a Google Doc or Google slideshow, or they can use their iPads and use the stop motion app. I think that would be really fun to have them create the graphics here. So I'm not going to set the timer, but I'm going to have you guys go ahead at your homes. Those of you who have this kit, you might need more space because you're going to be lining up all the chromosomes and separating them out to be able to demonstrate these 4 stages of mitosis here. So I'll give you just a couple minutes for that. Now, what's nice about having students do this in the classroom is you can actually see them and where they're lining things up. One other sort of classroom tip that I have is if you haven't purchased markers that write on the tables, this is a great activity for that, because you can have students draw the outline of the cell membrane on the table, maybe as well as the nuclear membrane, and have them place or move these chromosomes in the different sort of cell outlines. And you can have them label it, or you can have like big chart paper, have students put it on there and use markers and draw the different stages and take pictures. So definitely very kinesthetic. And of course, with meiosis, as we're going to talk about that next, it's like a huge diagram. And so that could be really fun, like lay it on the floor, line them out, have a definitely them modeling these processes. Okay, so your students, you might have them do a stop motion video. We had some of our 3D molecular design interns do the same thing with stop motion. So we're going to show the next video of their version. All right, so you can see this video as it's playing. Let's go ahead and play a couple more times. This is the classroom kit with 6 chromosomes. So the student kits have 4 and the classroom kit has 6. So you can see 2n equals 6 there. You could see the students can put on the kinetochores and spindle fibers to be able to show where those spindle fibers are used as well. One little thing I have students do so they know that what you're seeing there is actually part of the whole cell cycle, that it's G1, S, G2, and M is, if you can see my camera, I have them say, this is a chromosome during, not exactly, but it's G1 is by itself. And normally it's condensed and we'll say, this is chromatin in G1. In S, you say this is a chromosome replicated because that's where the DNA gets copied. And then you're going to say this is still chromatin in G2 because it hasn't fully condensed, but you have the duplicated chromosomes. And then you can then go into prophase, metaphase, they all line up. Anaphase, they separate, and telophase, they're in 2 different nuclei forming. So you can use this to go through the whole cell cycle in the context of the life cycle of a cell and not just mitosis, because I think a lot of kids in the S phase don't recognize or realize that's where the DNA gets duplicated and that's where DNA replication comes from. Okay, so moving on to the next slide. With students, you can kind of talk about the number of chromosomes before and after, what are the cells like, genetic material, and then how many chromatids were in the cell during prophase, and how many chromosomes are in each daughter cell. So the idea here is your students get to see, you start with 4 different chromosomes, or these 4 chromosomes, and the daughter cells you end up are identical with these same 4 chromosomes. Okay. All right. So now asking you to put your teacher hat on in the next slide is how do you think this particular modeling kit might be useful in your classroom? What do you think are some misconceptions or aha moments for students that are going to crop up? So feel free to share that in the chat now. Would love to hear your thoughts. And then Chris, if you could click on to the next slide. There you go. Yeah. So I'd love to read or hear what you guys are thinking. How do you think this kit might be useful in your classrooms or with your students? Okay, thank you for responding. Cassie, great to get hands-on experience moving the parts. Will help them understand it, definitely. Elisheva, clearly visual representation of the process is ideal. And then Lucy, I like the way the pieces stay the same throughout the different phases, different forms of the same genetic material. Yeah, definitely. Now I do have to mention that before this, I used some different materials to model mitosis, which are helpful as well. I've used like yarn, and I've used pipe cleaners with beads to represent like the kinetic of the centromere, like where they're attached together. I've used socks, like I had a set of I attended a Chromosomes presentation. I used felt, like I cut it apart and had like felt pieces that were long or short, and even wrote like the genotypes, like big A, little a on them. And certainly you could use those as well to model mitosis. But you're going to see as we do meiosis that this particular kit is really unique, that is far better than like socks or pairing socks or pipe cleaners and such, and which is helpful. So yeah, and then Lucy was talking about challenge to put yours together and thought it was a good time to talk about handedness and lateral symmetry. Lucy, do you want to say a little bit more about that?
I just think it's, I mean, it's such an important thing in biology, and it's hard. Is it the same or is it a mirror image? And that seems like it's still mysterious to us, how we end up being that way. And it's not necessarily something we realize clearly as we're going through things. We think, you know, that mirror image is the same.
Ah, okay. I hadn't thought about that before. I appreciate you mentioning that. How it's a right-handed helix. Is that what you're referring to?
Yes.
Okay. One thing that's really cool is, like, you know, right now we're showing a 2D static, you know, flat, how chromosomes always been represented this way. And you would think that the identical sister copy is just a a mirror image, and, you know, kind of like your hands are mirror images, but they're not quite the same. You could also have students do this. You have this, and then you put it right on top. And so you know that they're identical, you know, both in, like, I guess if you think about it, the direction. So it really, maybe you brought up a really good point that it shouldn't be flat like this, where kids get the misconception that these are mirror images. But rather than, you can, because it's now 3D, you can layer them on top and say, no, they actually are identical like this. So I think there's that you could have students do that as well. Same thing with when we're going to do meiosis, that you can have them sitting on top like this, so that they can see that in 3D space. I appreciate you bringing that up. Thank you. And then Michael saying, again, that students have hands-on experience, that it's a game changer. Absolutely, absolutely game changer. Okay, so now, so I appreciate you guys sharing some of your thoughts on that. Now we're going to go to, like, more advanced level. We did cell division with making identical sister copies. Chris, if we could go back to that original meiosis slide there, good. Now we're going to go through meiosis, which is needed for sexual reproduction. So sexually reproducing organisms, in order to reduce the number of chromosomes, so that when they combine, eggs and sperm combine together, we have back to the original number. Otherwise, if you have a human cell that's 46 with a human cell that's 46, you're going to get 92, and then so forth and so on. And that's going to wreak havoc in the cell in terms of regulation of genes, gene dosage. And so it's great to reduce the number of chromosomes so that when they combine together and have a lot of genetic variation in that recombination, you have that same number again. So process of meiosis actually allows organisms to increase their genetic diversity in several different ways. And we will come back to this idea of genetic diversity again. But for now, notice this graphic shows 2 divisions that, again, we still have the 4 chromosomes in there, the original cell. You're still going to have cells go through G1, S, G2, and then now you have this 2 cell divisions in the M phase. And you will see that there's meiosis 1 and meiosis 2. So at the end of meiosis 1, you have 2 cells, and at the end of meiosis 2, those 2 cells divide and become 4 cells. So you have 4 daughter cells. All right, so we're going to model this, and the next slide will show, as a reminder to students, going back to that cell cycle, you have the 4 chromosomes that you'll have, or 4 DNA molecules, you could say, in G1, and then in G2 or S, their chromosomes replicated. And so going with Lucy's idea, these aren't mirror images, you could just have them lay them on top of each other like this. And just to show it's two identical copies. They are connected together though. They are connected together at the centromere. And they're going to stay together for a special reason. So you could have them replicate there. And G2 phase, we're not seeing anything really change about these chromosomes. And now we're going to go into meiosis. So this is the sort of complex version of what's happening. And Tim Herman, who's kind of developed these activities, he wants to know what I'm going to say about these. And I'm essentially just going to say that meiotic crossing over is pretty complex. You can see in this diagram of how there's double-stranded breaks, and there's something called strand invasion, where you have the homologous chromosomes are next to each other, and they're going to do a little mix match, sort of a mixing there, and where they're gonna, one strand is gonna invade another. There's going to be some breaks happening and then repairing as well. So this is kind of a complex process. You probably aren't going to go into this with the middle school level. I know there's some middle school teachers out there. At the high school level, if you've got AP kids and they're asking the hard questions, this next graphic or image is going to be really great to show because it shows sort of the more complex stuff happening with holiday junctions where you have the two strands coming together, the breaks, and sort of the crossing over happening. So the next slide, let's go ahead and watch this video. So let's see that again. Did you see how those two different how there's the homologous chromosomes, they're super close to each other. There's going to be invisible enzymes that we're not seeing in this that dictate, the combining of these molecules, the cutting of them. You're going to have nucleases and probably polymerases, like doing that repair, which is what you're seeing with the gray. And this would kind of be great for students to see, like, these are some places, this is a place where you can have some genetic mutations occur during crossing over, and why you may have, non-disjunction occur, or like pieces of chromosomes that separate off and attach improperly somewhere else because there's actually this breaking of chromosomes here. The whole point of crossing over is actually just to ensure that these homologs align properly at the metaphase plate and are attached properly to the spindle. So they're actually still kind of attached together at this point in a group of 4 chromosomes called a tetrad. And so it's kind of crazy to think that they're not mixing and matching just for the sake of mixing and matching, but it's a mechanism that's evolved to be able to ensure these chromosomes are properly aligned together, these homologs are aligned, and that they attach to the spindle. So that is the sort of the hand waving I'm going to say about this. I do want to give a shout out if you would love to learn more. 3D Molecular Designs with the Center for Biomolecular Modeling offers teacher workshops in the summer in Milwaukee that will deepen your biology knowledge and content knowledge of all of these processes. And so I didn't learn about this that you're seeing in this video until 2 years ago, and I was just mind blown on how this occurs. So if you are interested in doing some professional development and would love to learn more what's happening at the molecular level, I highly encourage you to sign up for one of these Center for Biomolecular Modeling courses in Milwaukee. And thank you, Rachel. This specific topic is from the Science and Ethics of Genome Editing, which incidentally, I was part of a group of teachers that got to do our workshop in Berkeley last summer. And we got to do a whole week in the same biotech building where Jennifer Doudna's lab is. And We never got to meet her in person, but we saw her walking with her coffee cup past our window every day, past our conference room, and would like kind of wave and, you know, like drool over her and be like, that's Jennifer. And the closest we came to is a couple teachers ran into her in the bathroom. They were so like overwhelmed with seeing her. All they could think of to say was, hi. And then that was it. So that was our Jennifer sighting at Berkeley, but we did get to visit her lab and see all the neat projects that they're doing related to gene editing. So very cool. All right, so let's go ahead and go to the next slide. Let's take our chromosomes and do crossing over. So in prophase 1, we have a couple of major things happening. You have during replication, these sister chromatids stayed together, and you may at this point want to just kind of put them on top of each other like this, so that in 3D, in this 2D image, you're seeing them like side by side. In reality, they are on top of each other. And what you can do is some strand invasion and the whole breaking and swiveling and swapping pieces. Now, this is the power of this kit, is I can sit here and go, I'm going to take off this part of the red and this part of the blue, and I'm going to swap them so the red goes on the blue and the blue goes on the red, so that you can see here like that I have these mixed pieces now. This is the crossing over that occurs. This foam kit allows you to do crossing over at a couple of places. So in actual crossing over, it will happen from 1 to 3 locations. So maybe you could do it at another place. So cut and swivel and swap. And so that you have these new chromatids that have, you know, a mixture of blues and reds, and that is crossing over. I have not been able to replicate this kind of thing, simulation, with pipe cleaners or socks, or I had a really great like physical activity where students got playing cards and got to pretend they were chromosomes. But I always ask them, please don't rip my playing cards and swap pieces like in crossing over, because I wanted to use my cards again. So that was a limitation of that particular model, and like a pipe cleaner model or the socks model, is you can't actually swivel and swap. And I love that kids can swivel and swap wherever they want on these foam pieces. So if you have these kits at your house, go ahead and try, you know, this crossing over with both sets of chromosomes there. And so I'm going to swivel and swap these parts right here. And this is where you can see you could have some mutations occurring too. And students may on purpose do that too. And this is where you can see as you're walking around the classroom, if you have a classroom kit, how students may not be understanding. And you can easily spot where someone's got, you know, some kind of chromosome mutation because they reattached, you know, a short piece with a long piece. And then you've got these unequal sized chromosomes. So then you can kind of praise them and say, oh, wonderful, you've skipped ahead to showing how genetic mutations can occur during this crossing over. However, we're going to show what normally should occur, and then you try to have them rearrange it back. So this is where you can, you know, and students sometimes at that point are like, oh, I can see how easy it is for these mutations to occur. And that is one place where you can introduce genetic variation right there. Okay, so I'm gonna give you, probably don't need 10 minutes, but go ahead and do your crossing over, and then show on your tables, maybe physically you show the moving of these chromosomes so that you can kind of get an idea of what are students gonna do and how might that look in your classroom, how much space you might need. That's pretty helpful too. So go ahead and do the student role and give you, yeah, so maybe, so Chris, you can start that timer, maybe go down to 5 minutes on that one. Okay, let's see how you guys did. This is a really important time, like for teachers, as you're circling around the room to see how students are doing meiosis. And because there's a tricky part in metaphase 1. So next slide will show how those chromosomes align up at the metaphase plate are what I call like Noah's Ark formation. They're 2 by 2s, and so they're going to go in pairs. And the way they're arranged on that metaphase plate could be in this orientation where you have 2 maternal chromosomes on the left and 2 paternal chromosomes on the right. And then anaphase 1 is where I call chromosome divorce, where those homologous chromosomes separate and their sisters are still together, but the maternal and paternal chromosomes now are separated. So at the end of telophase 2, you have these 2 daughters, or telophase 1, sorry, you have these 2 daughter cells that are already haploid. So we started off with 4 chromosomes at the beginning, and you're already haploid, or half the number of chromosomes at the end of telophase 1, that first division. So no longer are they same or identical to the parent cell. They have half the number, so you can see 2 chromosomes in each of those cells. And then prophase 2, metaphase 2, anaphase 2, telophase 2 look very similar to mitosis. So that's the comparisons there. Really the trickiest thing to watch what students do is metaphase 1 and anaphase 1. Do they know how to separate those chromosomes? All right, and then can someone else in the chat type in where, besides the swapping of the pieces with crossing over, where is another place of genetic variation that you see in this diagram that could occur? So the swapping of the chromosome pieces, okay. All right, Trinity's typed in how the chromosomes line together. Trinity, would you mind sharing a little bit more about that?
So how they kind of, the divorce, right? Like the divorce part, how they're lined up after they have that separation will determine what cells they end up going into, right?
Right, yeah. So what's interesting is I think in this diagram, and I didn't create it, I think one of the interns did, was you can see metaphase 1, they were lined up maternals on the left, paternals on the right. Now you can see in anaphase 1, they had like flipped. So that's like another variation right there that you could end up with, how you end up with the daughter cells. So exactly right there. Thank you. Okay, so moving.
Can I pipe in here and say something, Chris?
Sure, Tim.
Because this is what I was sort of alluding to with my chat comment earlier. Because to me, this is an underappreciated point. And it's just because I'm not a geneticist and I never really have taught this subject. And I had always heard about this random segregation of chromosomes. I think that's the term that I remember hearing one time. But I never really appreciated how important it was. And from my point of view as a reductionist biochemist, you can forget all about the crossing over as a source of diversity, because you can get plenty of diversity in offspring just through the way these chromosomes line up during meiosis 2. Because you can make, I think the calculation is with 23 chromosomes, you can make about 8 million different little people with different genomes, because you're mixing up 23 different chromosomes. You take a chromosome from mom or from dad in the new offspring. So I have grown to appreciate the significance of how these chromosomes segregate during meiosis. To me, that's been a new realization of what's going on during meiosis that I had never appreciated until just a couple of years ago when we started working with all of these. So.
Absolutely. Thanks, Tim. I think that would be a great point for your students at the end, if they're doing this, whether it's at home or on their tables at school, of say like, okay, you grab one cell, show it up, what their chromosomes are in that cell, like maybe the rightmost cell at the end of telophase 2 is what's the genetic material that's in that cell, compare it to somebody else. And you can see that probably in your classroom, there's no two that are necessarily alike, depending on if they had crossed over or not. So that's pretty powerful to be able to use this model for that.
Yep. Okay. Well done.
Okay, thanks, Tim. Tim is the godfather of all of these models, by the way, if you guys have not met Tim Herman. So appreciate his. All right, so if you have your students do this at home, they could make stop motion videos and show this claymation wise. So let's take a look. So you can see there that chromosomes, the pairs line up like Noah's Ark 2 by 2. And then they segregate independently, and then they go through all the different stages of meiosis. So I would say that for this model, having it static in 2D of these chromosomes next to each other is probably a disservice to the use of this model. And like Lucy was saying, like maybe just laying them on top of each other. And that way you can see that crossing over happen in those homologs lining up together. Great. Okay, so thinking about questions you may ask your students. These are some common questions, how comparing the beginning and the end, how many chromosomes are there, how many chromatids. You could ask them at the AP level, maybe diploid or haploid, talking about the 2N number and the N number. And then comparing the process of mitosis and meiosis. How are they similar? How are they different? There's a lot of discussion that come from this. So thinking back to the ways in which DNA was being mixed or shuffled up, we have already mentioned the crossing over. We've mentioned the independent assortment. Now, what's another source of genetic variation that you may want to bring up with your students in terms of sexual reproduction? How that is a benefit for species. One thing that's fun to talk about with kids, because it makes them uncomfortable, but it's a natural part of life, is sex, right? So there we go. Thanks, Trinity. So random fertilization with those eggs and sperm cells. Can't choose the gametes that combine, so thank you. Yeah, it's a surprise. So when we talk about sex, have that combination of eggs and sperm, and that initial cell that's formed can have a combination. So, and that's what Tim alluded to as well. So thanks. Okay, so that is really the power of these kits. Just want to give a minute or two here of what are some ways that you might be able to use this in your classroom. And for those of you who've tried some other kits before or different ways of modeling mitosis and meiosis, how might this be helpful for your kids? Okay, thank you. So once our kids come back, this would be a good way again to visualize the randomness of each one of us. Yep. Yep, Trinity, I agree. Like pipe cleaners, beads, string, Play-Doh, they're fun, they're great, but there's some limitations of those. And so, yeah, I agree. Seeing this kit, I was like, I'm gonna put my my other stuff. I had some felt chromosomes and all that went away. It's like, this is not going to replace that. This is going to replace all those things. So yeah, absolutely. All right. So thank you for sharing. I hope you've seen how useful this chromosome kit is. Now I'm going to include in the last 9 minutes here, kind of a little bonus lesson that you can add with this. So we're going to play a role, and you can have your students play this role of being a genetic counselor, and going to introduce you to a lovely young couple, Mr. and Mrs. Smith. They're getting ready to start a family. So let's talk about a very common case that we utilize in our classrooms. And so we're going to go talk about the story of beta globin. And so the HBB gene encodes for a beta globin protein. And as you can see in this diagram, within red blood cells, there are hemoglobin molecules that are made up of 2 subunits, 2 alpha subunits and 2 beta subunits. And the main job of this protein is to be able to carry oxygen for these red blood cells. So you will see there are 4 heme groups in there, and within them is iron. And so that's what makes our blood red, why you have to have, spinach and be like Popeye. And I don't even know if kids know who Popeye is anymore. Probably not. Maybe. I know I'm having to update all my references because kids have no idea what I'm talking about when I do these cartoon or movie references. They get younger, I just get older, or they stay the same and I just get older. So anyway, so in this HBB gene encodes for the beta globin subunit of this protein. And so it's pretty important to have this in your cells, in your red blood cells. All right, in the next slide, you'll see that we're going to go ahead and zoom down for kids to look at the molecular level. Where is the phenotype of what we're going to talk about, this genetic disorder, coming from? So we're going to zoom in. The HBB gene is located on chromosome 11. So we're going back to this karyogram again. And then the next slide, we're going to zoom into that chromosome, and you can see it's located on the P arm of chromosome 11 at locus 15.5. Okay. All right. So as we take a look at and zoom into the DNA sequence of this particular gene, there can be a spot where there's 2 variants, and we call those alleles. So what do you see are the differences between these 2 spots, or these 2 loci? And we've modeled them with our foam there. So this is kind of like a Where's Waldo moment. Can you spot the difference? Okay, so you can see that A's and T's are swapped, and we kind of want to take a look at that bottom strand where you're seeing on the left CTC and on the right is CAC. That's what is our template strand. And so in the next graphic, you're going to see what that difference will result in. So on the left is that CTC that we mentioned before, that T, and then on the right, you'll see CAC, a single nucleotide substitution. So, and so that's listed, in SNPedia, where they have the gene sequences of this. This is the most common mutation of the HBB gene at this location. And so when we go look at what will that cause in terms of the protein that it encodes for, this beta globin, you'll see that the RNA sequence for that left side, that CTC will transcribe to GAG in the mRNA. And when it's translated, the ribosomes will insert a glutamic acid in there. And glutamic acid is hydrophilic. So in terms of properties of these side chains, glutamic acid is hydrophilic. That means they're water-loving and generally will fold and point to the outside surfaces of the protein. Now, on the right side, we have a mutation. It's called a missense because that CAC, where you have a substitution there, will transcribe to GUG and translate to a different amino acid called valine. And valine has a different chemical property than glutamic acid. It is hydrophobic. So it changes the properties. And in biology, one of my favorite quotes to always sort of have kids be their mantra is structure dictates function. And if you change the structure, you're going to change the function. And so let's take a look at what's going to happen there. And so in this protein, on the left, that's our normal hemoglobin, and that glutamic acid is going to stick out on the outside because it's hydrophilic. And on the protein on the right, you'll see this is what forms an abnormal hemoglobin. And it kind of looks sort of the same sort of shape, but because of that valine on the outside, it makes it, that's a hydrophobic amino acid. And so it creates kind of a sticky pocket right there, which means other hemoglobin molecules will stick to that as well. And so in what can happen is you have all these sticky little parts, and that creates this clumping effect in a linear fashion, as you see on the right. Okay, so I know we don't have a ton of time left. I apologize for going more quickly through this. And so the variants that you're going to see with these abnormal hemoglobin on the right and normal hemoglobin or beta globin on the left. And for kids, what you want to lead them through, and the next slide shows that you can talk about how individuals can inherit different genotypes because of these different alleles that they could inherit. And the next slide, connecting that with genotypes and shorthand of things like big H and big H, or little h, little h. And the next slide is asking kids to think about knowing this about protein folding and the stickiness or not. How might that affect cells? So next slide. And on the outside, like in terms of function, what you can see is that the red blood cells are roundy shaped with normal hemoglobin molecules, and because of that clumping, distorts the shape of those red blood cells and make them sickle shaped. And you can talk about how the normal shaped cells gives you a normal phenotype, and the sickle shaped cells, due to that mutation in the hemoglobin and beta globin gene, could give you abnormal cells and cause sickle cell disease. And if you inherit one of each copy, you get sickle cell trait. So on the outside, you appear pretty normal unless you're in certain situations where you may have low oxygen levels, and that might tend to show up. But for most part, they're pretty healthy. So as you go through the final few things, this shows sort of what happens with sickle cell trait. Can cause a lot of physiological problems in patients with sickle cell disease. And now you go back to the Smiths, and you can model for the Smiths or talk to them about giving them a blood test. The next slide. And finding their carriers. And then what does that look like in their DNA? And you can see that that's what it would look like in their DNA. And then keep going. And you can use a Punnett square and have students model the nucleotides and have them put them into possible boxes like the Punnett squares and take pictures of them there, and to show what the resulting genotypes could be, and ask students to figure out what would the phenotypes, would they be normal, would they have sickle cell disease, or would they be carriers and have sickle cell trait? So finally, at the very end, we're going to kind of skip to the very end, is this gives your students an opportunity to talk about the ethics of genetic counseling and what implications that might have for this family, and to make informed decisions about health outcomes. So, all right, it's the end of our time here. Here's our connections with NGSS, and I'll stick around if you have questions or want to ask 3D molecular designs about purchasing things. But thank you so much for allowing me to spend your morning with you.